Yeah, and I'm really going to focus on the aspect of simulating microbial ecology. So not really, yeah, the numerical analysis or the analytical part, but really how do you simulate a microbial system? Just a second. Yeah, and then the first question, of course, you might naturally ask is why do we want to simulate such a system? And yeah, we know that at a microscopic scale, microbiomes are fairly complex. So there's lots of things going on. So if you think of cell-to-cell interactions or fluxes between cells, we have these diagrams, but in reality, it's a very complex picture, if you think. But when you look at a macroscopic scale, you usually see some fairly reproducible patterns. So here in this example, you have like a tumor tissue, and you have a healthy tissue, and you can see that, well, communities sampled in the tumor tissue cluster compared to communities sampled in the healthy tissue. So you see these macroscopic kind of patterns that you can say they're kind of emerging patterns. And you want to know like which components of the microscopic scale are important to explain these microscopic patterns. I mean, for sure you cannot get all of them. So that's the basic question. So what are the essential or minimal microscopic components that you need to reproduce the microscopic patterns? And you can think of this like an exercise. So you're like playing the tape of ecology multiple times because you're doing a simulation in a computer, so you can do it multiple times. And you make microscopic changes to, you make some changes to these microscopic components. You choose which components to use. And you see how that changes the variational landscape of the microscopic patterns. And you see, well, if you get, some intuition of how they work or why they should function. But of course, you can ask maybe a more fundamental question is like, why do we think we can do this at all? Second. And yeah, one of the things that we can all agree about microbial systems is that, of course, microbes live in nature in very large numbers, in much larger than, for example, of course, when we talk about macroecology. But if you think even in every community, you have billions and billions of cells. And a simple argument would just be that by this law of large numbers, you would expect to observe sort of patterns, that things would converge to some average. But not only this, like if you think of biological systems, so biological systems, they have this hierarchical nature. And one thing of this hierarchical nature is that, well, when you look at patterns at the higher levels, they should be constrained by the ones at the lower level. So that's like a general expectation. Maybe you cannot predict patterns at a higher level from a lower level, but at least they're constrained by the lower ones. And an example, a very classical example, if you think of microbial growth. So like Monod in 49, showed this very nice relationship between the consumption of glucose and the growth of cells. So if you think microscopically, a very complex thing is going on. So you have, consumption of glucose, at least more than 12 reaction steps to grow, to produce energy and to grow. And yet, you can find a very simple relationship between the number of divisions per hour and the amount of glucose that you have in the media. So the idea is that you get patterns, they emerge from a few or simple microscopic features of the system. And of course, the motivation that I'm doing this is that, well, we built this R package that's called MiaSim that Deolati shared in the chat, so you can access it. And is a package that allows sort of microbiologists and users to very simply have access to ecological models and to build their own simulations and try to test these patterns with their own system. We're gonna show the package this afternoon. And it was developed in cooperation with all these people that are shown here. And then, so we start like some questions that you can ask about microbiomes or about species in general is what maintains species diversity, why some species are more abundant than others, why some species are found in one environment but not found in others. And there's this classical view that kind of, you know, one of the first attempts to explain this in a very general way is that, well, organisms have this kind of a niche that's a space of where they can live in. And that's kind of a projection of, so you can say, well, maybe genomes evolved to different possibilities, and you can more or less project that into reality and say, well, there's this range of where an organism can live in, and you say this is like a niche. And if you think on interactions, then you can like maybe get all of these conditions, collapse them into one dimension like this, like do a dimensionality reduction or something. This is, of course, just an illustrative exercise. It's not a real, I don't think you can really do this computation easily, but you get all the conditions of where a species can live in, you project it into one dimension, and then you say like, where would a species fit in there? And then you would have like what's its preferred range, its preferred conditions, but also the range where it tolerates. And basically you say, okay, each species has this kind of this niche. And based on this niche, you can maybe predict what a species abundance, or what is the actual condition in an environment, and then what would be the species abundance in this environment. So it'd be something like this. So let's say the environment has this condition over here. Then you would predict that well, species A would grow in it. Species B can live in it, but then it would likely be outcompeted by species A. So where there's this overlap, one species outcompetes the other one. But you can have something like this, that where you would maybe have coexistence. So one species would be more dominant than the other one, but you would have a still coexistence. So this is like a classical view of how and why you would get and when you would get different species. And of course, there is some evidence. You can say maybe there's some there is a case for this niche view. So this is an example from a colleague. So that he clustered all the microbiomes he could find. So there's thousands of microbiomes here. It's corrected for some sampling biases. And you can see some patterns in distribution that they, you know, seem to be niche driven. You see that the host of microbiomes cluster, you know, microbiomes in saline and non-saline environments cluster, and in saline environments cluster, and you see these, you know, more tight clusters. So this seems to make a case for this niche view of the microbiome. And there's this recent paper where they did kind of the same exercise, so clustering, but now with metabolites instead. So you're going directly with the molecules. And this is a very, very simple plot. So it's just the richness. So it's the number of bacteria or of taxa in one environment, and the number of bacteria-related metabolite or microbial-related metabolite in an environment. And even here you see there's kind of this characteristic value. So you see that some biomes have, you know, a characteristic kind of number of bacteria and number of metabolites. So this again sort of makes the case for this niche kind of view. And you can, and the nice thing is that this is like nested, so it's a nested structure. So if you zoom into one of these environments, so these are like the biomes. So for instance, if you zoom into the human gut environment, you also find these kind of clusters and groupings that you know could or could not be evidence for this kind of a niche structure. So you say, well, there's the environment creates like a landscape like this, and around this landscape, a community would be formed. But of course, everything I said until now is like purely hypothetical. and you want to maybe, test it and assess it and say, well, what will happen if we relax this assumption? What do we say that there is no niche selection? And relaxing this assumption, so saying, well, we remove the effect of the environment on the microbiome, you can think of it in different scales. So you can say, well, first, you may assume that there is it's biologically illogical. You can say, okay, there's no niche effect. But of course, you would say, okay, you have anaerobes, aerobes, so you still have some. So you can maybe just zoom in into a particular environment. So you say, well, if I'm in the human gut, then the human guts are so similar to each other that you don't really see a strong niche effect in delineating. So you have a general filter. These subset of microbes can live in the gut, but then within these microbes, you just there's no strong environmental filter anymore. Or you could zoom in to a finer scale and say, well, if we go into a particular region in the gut, so for crypta, for instance, and here, the conditions are so similar that if you find a diversity of microorganisms there, they're not really selected by the niche. They're there because, for other reasons. So this would be something like this. So you would have it at a certain area on this space. You would have kind of several bacteria or several different species that have the same niche, and yet they're still a species, and they still you can still differentiate them molecularly or something like that. And they still live in the same environment, so they're not. So you would say, well, if with this niche field, if one has a slight advantage over the other one, then they would outcompete, but they would still live in the same environment. And here's an example. So it's another interpretation of the Goldford paper that Itzvan mentioned. So it would be something like this. So basically, what they did here is they gathered several communities from environmental sources, and they grew them in the lab in a glucose minimal medium. And then they checked for the composition over time. So for the like a stable composition. So they did several passages, and after several passages, what is the composition? And of course, across communities, you see like a diverse composition. I guess this would be expected, even though if you go at a higher taxonomic level, you see there is kind of a consistency between the taxonomic distribution. But the interesting thing is if you look into replicates, so this is like communities that were generated with exactly the same source community, and then replicated until stability. And then if you look at a lower taxonomic level, then you see that several different like compositions, stable compositions emerge. And if you look at a higher taxonomic level, then these compositions are stable or consistent. So you can interpret this that, well, there's several species that are from the same, like if you go for a higher taxonomic level, then yes, there's an environmental filter there. But if you go at a finer scale, they're basically equivalents. And then what ultimately dominates in the community is, well, maybe the product of some random or stochastic processes. And that's where you could maybe use a neutral model to say, so how would you then generate these communities where different species coexist, but they are basically ecological equivalents, like here. And the idea here, so I'll explain a bit Hubbell's model. So it's I mean, there's the general assumption is just that species are ecologically equivalent. You can think of several models that have this, that satisfy this assumption. And in this particular case, you would start with so it's very important that you say, the dynamics in the local community is not what really matters, but you need to have a meta population. So you need to have like some source community. So from where species come from. And from this source community, you define a probability distribution. So there is a probability for a species to colonize a local community. And then you start a local community and you impose a dynamic where you choose an individual at random. So with uniform probability. So this dynamics is at the level of individuals, not at the level of species or at the like at the levels of like you would have in a dynamic, in an ordinary differential equation model. You really look at the individuals, and then with probability 1 over ni, so ni is really the total number of individuals in the community, you remove an individual. So you do this kind of this Moran process, and you replace it with either an individual from the, I'm sorry, I don't know how to take this, just a second, with either an individual from the meta population, and with the so you have this probability that's, sorry, you have a probability that's mi, so that's the probability of migration, that's specific for each local community. So you would replace that with an individual. So you would get an individual from this distribution here. Otherwise, you would just select, again with probability 1 over now n minus 1, an individual to replicate from the local community. And then the species richness within a community, so how many species are within a community, will depend on the dynamics. So that's not something given. What is given here is the number of individuals. So the number of individuals is fixed throughout your simulations. And you can summarize one parameter, since you have different local communities, you can summarize them with like the likelihood of migration. And you can see here that if this number, like if you say that migration is 1, so of course, each time an individual is replaced with an individual from the meta community, then your community would trivially just be the whatever you define as your meta population. If this goes to 0, then the dynamics would only depend on sampling within the population, after, depending on how many species you start off with. After a long time, you would just go to one species will dominate the community. And the idea is that then communities live in a transit in between these two states. And you can, like, so it doesn't trivially go to your meta community. You can also introduce a speciation rate, so at maybe a much slower scale. There's some chance that a new species would be introduced into the community. And the idea here is that your meta community, so if you think of this meta community as some kind of fundamental distribution, so that's something that's given, and you draw samples from this fundamental distribution, and the local communities are empirical distributions that emerge from your dynamics. So you can think of this like this Dirichlet process. It's basically a Dirichlet process. So you can say your dynamics is, well, you have some probability to sample from the meta community. You have some probability to get an A, to get a B, or to get a C. Of course, if you do this process, you have to say how you would limit this process. So you can repeat it, for example, until the size of the community. You could also relax the assumption of the distribution H. You can say the distribution H is also like a Dirichlet process. And if you do this, you get this hierarchical Dirichlet process. This is just a curiosity that you can recognize this process as a Dirichlet process. And another thing that you can do, you can also replace this time step process with a like stochastic simulation process. So you can, with a rate, and you do that with the so-called Gillespie algorithm. So you can use you can replace the probabilities with the sizes, and then apply a Gillespie algorithm, and then your dynamic would go like, okay, instead of a point process, you say, what is the time for the next event? And that's where you would then drive the system to the next event, and then execute each of these iterations. This is maybe just a curiosity, but this is how we implemented it in MiaSim. So it simulates kind of like a dynamic process. So you go to the next time point, and then and so forth. And the idea that you do this simulation, basically you have maybe three conclusions. One is that your community composition is possibly sufficiently explained by a neutral model. You might have it that is also equally well explained by a neutral model. So that might just be your statistical neutrality. So you don't really care if ultimately what generated your community is neutral, but you can, explain it equally well with a neutral model. You might have that your community is better explained by a neutral model than a niche model. Then that would, mean something, maybe that the process that drives your community is neutral. And often, because it's hard to decide between one or the other, you would just, recognize that neutral and stochastic forces shape the system. But, you can't really identify what is the driving force. And that's why you would sort of make a simulation and how you would fit the neutral model then. So to fit the neutral model, you can start from this assumption that if your dynamics is neutral, you should have something like an asymptotic curve where so if a species is highly abundant in a community, you would expect also that it would be frequent across community. This would just emerge from sampling. I think it's an expectation from kind of any neutral process. And then based on this dynamic, you can, so three things can happen in your system. So if you zoom into one species, so here I will zoom into species B, so three events could happen to species B. So what could happen is that it could be, it can replicate. So one of the species dies from the community, then it replicates. So you have B and then you go to B plus 1. You could go to B minus 1, so and then be replaced by some other individual in the community, or it could stay the same. So you have kind of these three possibilities, and described here, like what would be the probability for each. And then you make some assumptions. So you make the assumption that, well, first the numbers are large enough so that the proportions can be modeled or can be properly modeled by a continuous distribution. Another one is that your system is at some stationary state, so it's been sampled enough that it's stationary. And if you do that, you find that the system will be distributed like a beta distribution, a truncated beta distribution, and with these parameters. So what this means in practice is that you can estimate your XS, so your proportions, by the cumulative density function of a beta distribution, like this. And if you look at this, you have things that are usually defined already from your sample. So you have the number of reads. You have the probability that you establish for the meta community. So that can be something like maybe the average of species across the meta community. And the only parameter that you don't have is this M parameter. So you have one free parameter, and then this allows you to then fit data to your model. And this is an example done by Sebring and collaborators. And what you get here, you get basically two answers. So you can see if your system fits well to a neutral model, and that's based on this R square. So it's how well all the taxa fit to this neutral process. But you can also see which taxa deviate from this process. So here, for example, in the mice, the Shigella has a higher mean relative abundance across samples than frequency in samples. So that means that some samples have a high Shigella numbers. So you get these two types of answers, and here you're focusing into the if the dynamics of one species is neutral or not. And the other thing that you could do to test for neutrality is use this, like you're comparing to distribution. So this is the statistical neutrality that I mentioned before. So you want to know if, so you have a neutral system and you have some other system, and you want to know if they're statistically different, basically. And then you can use like a measure to compare distribution. So there's this Kullback-Leibler divergence measures is a possibility. And this would work like this. So you have some representation of your original system. And this representation, you need to be able to take samples. So, and then you would reconstruct the system with a neutral model. So you say, okay, you could fit maybe the model, and then you can replicate it, reconstruct it with a neutral model, and then you compare the two with this Kullback-Leibler divergence. And if this distribution is centered at 0, then you would say that you cannot differentiate the dynamics of your system from the neutral dynamics. And so this can be used, for example, to compare models. So one idea is that, okay, you have a Lotka-Volterra model, for example. If you add some stochasticity to it, then it's clearly not a neutral model. So species have different fitness values. Yet you cannot differentiate it from a neutral model at a certain parameter ranges. So if the interaction diversities are low, for instance. And yeah, you can maybe extend this idea to not only comparing models, but comparing like moments of your system, for instance, the average of your species composition. And then another kind of neutrality. So we first relaxed the mesh assumption, but we could also relax the interaction assumptions. We say, well, obviously species are not neutral with respect to the environment. I mean, different environments favor species differently, but maybe they interact so weakly that this interaction is not really, relevant, or they interact with each other so much that these interactions are not different from a random process, for example. And then you would ask, okay, what happens with a system where, okay, it's not neutral, but species don't interact in a meaningful way. And this is where you get the stochastic, the logistic model. That this model is going to be explained, I believe, in the next lecture. That's why I'll just rush through it. And basically, is you have logistic curves for each species that they don't interact, except maybe for the noise. So you add, of course, you add some migration, and you add a noise term, and then in the noise term, and I'm going to explain this noise term because it's the same that we use for all the other models, is that you can account for different things. So first you can think that there's like sampling error. So that would mean simply that each time you take samples from your system, and there's some error there. So this error does not propagate across the simulation. So it's an error that's independent of the simulation. You can have some intrinsic fluctuations. So you can say, well, I have this model with these parameters, but they're not perfect. So each time an event or something happens in this model, there's a deviation from this parameter. So that would be something like so there's fluctuation around your model parameters. This would be an intrinsic error. And you can think also that, well, the species have some fitness in the environment, but the environment, changes around a little bit. There's some oscillations around the conditions of the environment. And this would be an extrinsic kind of noise introduced to the model. And the important thing is the correlation structure of this noise. So if noises are correlated with each other in time, so the closer the time point, the higher the correlation. And this is the so-called noise color. But there's also the correlation between model components. So if you have structure in your model, so for instance, if a species is high for some external factor, another species might also be high. And that's where you could introduce some kind of interactions, but that are not different from noise. And finally, you have these perturbations, so things that happen either induced or not, that they could be, of course, periodic, like if you have a system with a daylight cycle. They could also be aperiodic, so a system that is subject to some perturbation, and targeted and untargeted, and also post or press, so you can have like something that happens continuously, or something that is just happens once. So for instance, you can be like taking an antibiotics for a period of time, or you can just take one dose of antibiotics or something like that. So this, sort of this noise model is implemented in MiaSim, and it's every time I refer to noise, you kind of have to sort out these different factors in your model. And this is an example where they show that, well, for several time series, microbial time series, you cannot very well distinguish the time series from a stochastic logistic model. So this is a model with no interactions. It explains most of your observations just fine. The same way, maybe a model with interactions might explain, but then the question is, do you need a model with interactions if you can explain it well with a model without interactions? Yeah, and maybe the weak point of this model is that it neglects stability. So this was explained very well. And you cannot answer things like how large and complex a community can be. And another thing is that it doesn't really give you any practical insights into how to control or engineer a microbiome, because I mean, you can basically, you assume that species could, I mean, of course, you have very simple terms, only that species could exist or not in an environment. And of course, then it's not a suitable model if you want to answer questions like, what are the limits to diversity? Why are some communities more diverse than others, which is obviously what we see. So this is where you would go to the generalized, the Lotkavol-Theta family models, where you would introduce now the effect of a species on another one here, like here, the effect of B in the growth of A. So if you consider the growth of A, you need to consider some direct effect from B. And when you do that, you get this interaction matrix. In the interaction matrix, you can think of several things in it. So it's a simple, but you can, complicate the structure. So you can think of interaction strength. You can think of sparsicity. So this would be the connectance. So how many non-zero terms you have of the diagonal. You can think of the distance between the diagonal and off-diagonal elements. So how strong is the self-interaction compared to the interaction with other species. You can think of interaction types. So if it's like positive to positive or positive to negative, so you can think of these cooperation, competition, commensalism, amensalism, and also the interaction distribution. So this becomes important. So you can think like if you sample, for instance, from a random uniform distribution, or if you sample from a Gaussian distribution, but you could also sample from for instance, a power law. I would try to show this example in the afternoon. So if you sample from a power law, then you say, well, you have one species that's a strong interacting species that interacts with a lot of other species, while the other species between each other don't interact very strongly. And that gives you, a very different dynamics than when you sample it, for instance, uniformly. And you can also think of nested interactions. So you can think, okay, there's a subgroup that interacts very strongly with each other, but then there are other subgroups where it interacts weakly with each other. So you can think of this kind of a nested kind of structure. And this is something that I don't think has been very well explored. So it's a field to explore maybe. And then I'll repeat this a bit from the that was already explained. So I'll just go very quickly. The nice thing is that based on random matrices, so you can derive a bound on your system, so on the number of species that can coexist. And yeah, so either the interactions are weak, like in the stochastic logistic model, there are only a few species, or the species rarely interact with each other, so the matrix is very sparse. So are the things that you kind of have to accommodate, and you can think of environments that would change accommodating for these different things. And this is an example, just applying this theory that if you either increase the size of your species pool, so if you have more species competing, or if you make interaction strength, if increase interaction strength, you get kind of unstable communities. So either some species are extinct from the communities, or they go into these oscillations. And the other thing that you can do is you can vary, you can think of this, okay, does this hold if we vary interaction types? And this was done by the Foster group. So basically, if you have a higher fraction of one type of interaction, do you still see the same stability pattern? And you see that the same stability pattern does hold for all the types of interactions that you put. And one interesting thing is that even though they hold, they're not equal. So one way that you can increase stability is by increasing negative interactions. So this paper is very interesting because it suggests an objective way where you could engineer systems towards more diversity. So one way would be to increase the amount of competitive interactions. So if you want to go from a simple to a more diverse system, you would increase the number of competitive interactions. And then you have this that was also shown by Sun of this universal dynamics. And this is saying that the idea here is that if you have the same bacteria that occur in a different microbiome background, if they would follow this kind of Lotka-Volterra dynamics, then you would expect that their relative abundance would be consistent when you compare the two. And you can do that then by comparing two things. One is the overlap. So if you look how much overlap there is between a community, and the other one is the dissimilarity. So how different the overlapping groups are between each other. You do and then when you do that, so you expect, if this dynamic is universal, you would expect to see something like this. So in region that is highly overlapping, they should also be highly similar. And this is a pattern that you generally observe as well. And as was shown, they do that for the microbiome. And you also see so if you do that, you can also fit a curve and get kind of this characteristic complexity value that tells you how big should a microbiome be in terms of diversity. And all the examples that I mentioned so far, they're mostly based on large random interaction matrices. And maybe the most compelling one, that's this universal dynamics, is also like, it shows that there is a universal dynamics. It shows that the GLV would follow this kind of universal dynamics. So if you increase the interaction strength, then you would expect this kind of universal dynamics. But it doesn't necessarily imply that the dynamics is the GLV dynamics. It's a bit of a catch there. And to do that, you would have to actually fit the GLV to real data. And the question is how you do that. So you could have these top-down approaches. So one is you could, build, so you need to build this interaction matrix. You could build this interaction matrix from the co-occurrence network. You could try to build it from the time series. These would be like top-down approaches. And these have, some severe drawbacks. One, if you try to do it from the co-occurrence network, this would be highly biased by indirect environmental effects. So if the environment is different, species co-occur differently. This would be a big issue here. If you try to do it for the time series, one of the things that you find is that the parameters are very condition specific. So depending on the like you could find a good interaction matrix for one time series, but if you slightly change it, you find another one, and they don't generalize between time series. And the other one is these bottom-up approaches, where you can either, like one idea is you have a community and you drop in a species and then you fit a model, then you drop in another species and you fit a model, or you could cultivate the microbes and then try to fit a model by seeing how much one affects the density or the growth rate of the other one. You could also grow them on spent media. So these would be the bottom-up approaches. And then people have been doing this for some time. And what we generally find is that, you know, it's usually not a very consistent result. So basically, so this is an example, it's an analytical example actually. So if you assume a type of interaction here, so here's saying that this species one produces two compounds, one that favors species two and the other one that inhibits. And if you change just the density of the system, so you start with low density and with high density, you basically predict two qualitatively different interaction coefficients. This is an example from our system. So we have two bacteria growing in a system where, you know, energy is not renewed. So there's one does not have mucin in it. So basically there's only some sugar, and they waste the sugar over time. And here we add mucin. So mucin acts like a continuous source of energy. And in both cases, so this can be viewed kind of like an emulated kinostat. And in both cases, you have that, you know, interactions, they change significantly over time. So you could say, well, changes because it's a batch culture, but it's not the case. So over time, what happens is that, like, one bacteria acidifies the environment, and that has a negative impact on the other bacteria. I'm sorry, acidifies its own environment, so it has a negative effect on itself. But when it grows with another bacteria in the late time series, then the pH has a positive effect on the other bacteria, and that is significant. So the interaction switches over time, basically. And, you know, one thing that you can do is you can test to see if your system would fit a Lotka-Volterra dynamics well. So this is another idea here from a preprint, where if it fits a Lotka-Volterra, then you would expect that if you vary the conditions a bit. So one of the things is that the growth rate, the ratio between the growth rate and the carrying capacity should be consistent across different interacting partners. So if you put one bacteria with the other one, and then with another one and with another one, this ratio should be constant. And so you would have then a positive correlation between the growth rate and the carrying capacity as maximum LD. And what you see is that when you have sort of environment poor, so harsh environments, you get a good correlation. But if you go to, so if you increase the carbon concentrations, you go to a rich environment, then you lose this correlation, basically. So it's, again, the case of being a context-specific dynamics. And then the question is, how do we address this context specificity? So one way is, of course, to change the model, and that goes into, you know, derivatives of the model. And another way is to try to add sort of mechanistic detail, and that is the motivation maybe for the consumer resource model. I'm not going to repeat it, so it's kind of similar to the model that was presented before. But here, of course, you can have, you know, consumption and production of substrates. You can add a dilution term, so you can emulate the kinostat, and you get now this resource matrix. And this resource matrix, it can drive the system to very different dynamics. So you can either say that species co-utilize resource, for example, or they utilize resource sequentially. Then you would have to introduce, you know, functions of sequential utilization, so they use one resource at a time. And then you have this energy yield from the secreted product. So you don't want to get this like energy loop. So where a species produces a product that has more energy than the one it consumed. So you have to, put a constraint on that as well. And you can also introduce something like trophic level. So you can say, well, some species consume favorably one resource and secrete a class of resource, while other species would prefer the secreted resources and so forth. So you can do that, of course, in a fixed structure, but you can also do that kind of randomly. So that's those are the variations, and of course the variations are going to impact significantly the dynamics. Unfortunately, I don't have time to show some examples, but maybe we can see that with the MiaSim. And the good thing of using the consumer resource model is that you can, use this idea that metabolism is kind of a unifying feature of microbial systems. So you can get several patterns from metabolism. And one example is here where they could predict from the genomes. So they get the genomes and they fit the consumer resource model, and you can predict very well the growth rates of the bacteria. So you can, so the idea here is that you can correlate independent information. So like the genome or mostly the genome with the environment. So then you could go from the metabolic network, like it's shown here. So an example of a small scale metabolic network. You can maybe coarse grain it a bit. And you can, based on that, build a simulation that emulates a consumer resource model. You can use for this either like you can individual based, you can make it like a Gillespie algorithm, or you can fit actually a consumer resource model to it. I don't have time to go very into details, but the take-home message of what I want to say is that, well, if you think back to the question, what are the minimal microscopic components that we need to reproduce, the macroscopic patterns of a system? And you basically have this, one thing is that you need to choose an ideal level of coarse grainability, and you want your system to be predictable. You want to have, you want to test if there's universality. You want it to be convenient and also to have control. And basically, you have all these levels of choices when you increase, and trade-offs when you increase the complexity. And because you have all these, different choices, and also because subtle differences, result in different dynamics, ideally you would be able to test all of them or to test many of them. And you want the models to be interoperable. So you want to be able to, compare them between each other. And that is, the motivation why we made MiaSim. So you can, to test a random dynamic, neutral dynamics. You can test a stochastic logistic model. You can do an individual-based model, or you can do a Lotka-Volterra. And basically, you can kind of compare them because they're in the same sort of namespace, the same variable space. And you can, what is comparable between them, you can compare them. That's what I had to talk, and thank you for listening.